Pfizer used the least sensitive way to detect the biodistribution of the RNAs and where it's expressed in the animal model, and the FDA didn't catch them at it. Okay, and when I saw that, I knew that we were in trouble. There were no rigorous, good laboratory practice control, which is the standard, reproductive toxicology studies done. And now we hear all these things about signal, let's say, about spontaneous abortions and fetal deaths and all kinds of things that have to do with reproductive health. All that should have been detected long before it was ever put in humans. Now we have a mismatched vaccine that is being administered repeatedly, and there are multiple lines of evidence demonstrating that that can cause immunosuppression.
Dr. Malone, thank you for joining us.
Likewise, my pleasure.
Earlier in the summit, Dr. Urso was up on stage, and in regards to the mRNA vaccines, he compared the spike protein in the vaccine with the spike protein in the virus as to being butter and margarine, with saying something like the body can't process it because it's so foreign. Can you touch on that a little bit since you're so involved?
Richard seemed to have, and I don't want to cast shade on him, confused the issue that I've been raising recently about the pseudouridine in the mRNA, and had inferred that I was talking about pseudouridine within the spike protein. Mm-hmm. The spike protein, as expressed from the RNA, from the vaccines, is subtly different from the native spike protein present on the original Wuhan virus strain, that being two proline mutations in the S2 region, which stays cell bound when the S1 is cleaved off and released and goes into the circulation. And so the spike protein that is expressed from the vaccine is subtly different with these two proline mutations, but fundamentally the same as the spike protein in the virus itself, mm-hmm. except, as he did point out with that Cell article, the levels of spike protein that are expressed are much higher after the RNA vaccines than is observed after natural infection, and it persists for a much longer time. I think what he was intending to speak of is this incorporation of pseudouridine, which is the basis for the Carrico and Weissman patent that UPenn holds and makes a lot of profit from. Mm-hmm. And I needed to provide some summary information, actually for the Paris Summit that I was just at, in a talk I give in Marseille, at Dieter Raut's in Marseille Research Institute and Hospital. And in that, I wanted to summarize both the mechanisms of action and the potential risks of the RNA. It's a different talk than the one I gave today. Mm. And so I really wanted to dive into the implications of incorporating pseudouridine as opposed to uridine throughout the RNA molecule. And I had some suspicions that as we've seen these adverse events roll out, particularly the evidence suggesting immunosuppression, broad-scale immunosuppression, particularly of T cells, in the multiple vaxxed individuals, the triple vaxxed and quadruple vaxxed individuals, I thought that potentially could be a consequence of the pseudouridine, which is not normally present throughout an RNA molecule. Mm-hmm. And so I did a deep dive into the literature, including going back to the original Carrico and Weissman paper. And the reason why they put pseudouridine throughout the RNA, the synthetic RNAs for the RNA vaccines, is that it would suppress the inflammatory response, in other words, the acute immune response against these RNA vaccines, which otherwise reduce their effectiveness. Mm-hmm. This is something that we'd seen and battled in the laboratory for well over a decade. Matter of fact, I introduced Carrico to this problem set when I invited her to a meeting in Annapolis that I had organized many, many years ago before she did this work. And so that's why they put it in, was to shut down the inflammatory response. But the problem is that one person's reduction in inflammation is another person's immunosuppression. And so as this now was coming to fore as one of the major adverse events, I thought I really needed to go back and look at all those data. And it turns out that in the interim time, there has been some work done on pseudouridine and mRNA, and we now know, in ways that we didn't know back then. That the incorporation of this altered uridine, pseudouridine, which is a normal biologic process that happens in RNA. It happens as the RNA is synthesized typically. There's a specific enzyme that does this. It's very tightly controlled because the incorporation of pseudouridine makes the RNA last much longer, and it is associated with, among other things, these anti-inflammatory characteristics, which basically particularly shut down T cell responses. And then we saw all of this kind of come together. We're seeing clinically in the highly vaccinated this failure or reduction in response in the T cell compartment. And we also see this in the bench in the detailed research about the effects of pseudouridine, and that this is something that is very carefully controlled by the cell when and where pseudouridines are placed to extend the length of time that the RNA sticks around and its anti-inflammatory properties. And what's been done with these RNA molecules that are being injected into all of us that are accepting the vaccines is that these products, as they polymerize them, they use an enzyme that just produces this long chain like a string of pearls. They try to jam as much pseudouridine into that as they can. And now we know that that is absolutely not normal RNA, that characteristic. And in fact, in normal cell biology, it's tightly controlled because it has these other effects on these key properties like your immune response and like how long the RNA stays to get out there. So then suddenly the cell paper that says that the RNA stays around in your body for 60 days or longer from actual biopsies, this isn't bench research, this is taking samples from humans. And Pfizer and Moderna have said, oh no, the RNA just lasts for a short period of time in your body. Now we know somebody actually did the research. It stays in your body for a long period of time, continues to make this spike protein, and it seems to be associated with, particularly with multiple vaccinations, suppressing T cell responses. And now as I dove into the pseudouridine story and all the data on that, it looks like these characteristics that we're seeing clinically may well be a consequence of this modification that is the basis for the University of Pennsylvania patent that Carrico and Weissman assert is what was enabling for RNA vaccines. This is why they've sought the Nobel Prize for this, and there's been advocacy for that. But it looks like that choice may have been a Janus situation, a double-faced or double-sided, good for one thing, but causing some of these other problems that we're seeing. So that's what Richard was referring to, and that was a long-winded explanation. I hope it made sense.
Let me summarize it, and then you can tell me if I'm wrong. So you're saying that the pseudouridine that they use within the mRNA vaccines, it allows for the spike protein to last longer within the body, but at the same time, it does not cause the inflammation that a regular spike protein would.
No, wrong. No. Because you're making the same leap that Richard did. Okay, the pseudouridine is in the RNA. The RNA makes the protein. So the protein is the protein. It has its own biologic problems. It is a toxin. It has a whole bunch of toxic activities. It binds to platelets. It opens the blood-brain barrier. It causes other problems. So those are all problems with spike protein natively. The pseudouridine affects the RNA that makes the spike. And so what you have with what we know clinically, based on that cell paper, is that those RNAs are sticking around in people's lymph nodes for 60 days or longer, continuing to produce spike protein. So now it makes sense that the spike protein is coming up at levels that are higher than are in the infected individuals, and it continues to be produced for a very long period of time, 60 days or more. They didn't test beyond that. So it's not that the pseudouridine is affecting the spike protein itself, but it's affecting the template, the RNA, that makes the spike protein, okay? And that RNA has key characteristics of immunosuppression, which the spike protein also has characteristics, as Richard mentioned, interacting with P53 and influencing inflammation itself and downregulating inflammation. But also, we now know that incorporating the pseudouridine into the RNA itself causes those problems. Did that make sense? Did I kind of sort that out?
It did. And what is the reason that they wanted the RNA to stick around in the body for so long?
They didn't. They didn't think it did. It was, they did what they did, Grieco and Weissman, based only on the thinking that, this is a long time ago. This is over 10 years ago. Research on pseudouridine has come a long way since then. But they knew that pseudouridine, if it was placed in the RNA, would shut down the acute inflammation after you get it injected. This is redness, swelling, pain, those kinds of characteristics. And that it would reduce this. They didn't think about its effects on that suppression of inflammation could be a bad thing, and they didn't think about the effects of pseudouridine on making the RNA last a really long time. That wasn't part of their thinking. That's why I said I went back to the original paper. But now we know those are characteristics of incorporation of pseudouridine, even in the normal biologic sense, which is highly regulated, let alone in this highly artificial sense, where you just have a polymerase jamming in pseudouridine willy-nilly everywhere it can into the RNA molecule.
That's fascinating. Were people at the FDA aware of this fact when they approved the Pfizer vaccine?
The literature was there. This gets into a key issue, and you may not remember, it's a nuance in my story here, but early on I asked for it basically in the fall of 2020 as these vaccines were really starting to come to fore. I asked for a meeting with Peter Marks, and I expressed my concern about the Pfizer common technical document that Byron Bridle had obtained from the Japanese. This is this famous set of documents that shows that the lipids go to the ovary and that the stuff goes everywhere and all that kind of thing. Okay, so I was concerned about the possibility, because I have good contacts at the FDA, that the people reviewing the dossier package didn't really have a background in this area. This is totally novel technology. And for an average FDA reviewer type person, they've never encountered this before. There are no prior examples that they could have used. And a lot of this is pretty kind of insider baseball, specialty knowledge. My sense from talking to him, and what he expressed was they have absolutely no concerns about any of these issues. And I should await their final decision because they have more data that they're reviewing. That more data is this massive data package that they tried to withhold from the public for either 50 or 70 years, and now they've been forced by the courts. But he said that I should hold off and let them do their review of all those data, and that nothing that they had seen caused any concern. One of the things that came out of that was that the FDA has checklists. They operate by checklists. And they say, oh, have you done this test? Have you done that test? Okay. And those checklists are a function of the type of the drug or therapeutic. And what they appear to have done is, because there was so much denial that this RNA technology was essentially a gene therapy technology used for vaccine purposes, they didn't apply the checklists that would have been normally applied for a gene therapy product. They only applied the checklists that would be normal for a traditional vaccine product, which this absolutely isn't. And so they moved down that checklist, and all the studies that they thought would be necessary for a standard vaccine were done to their satisfaction. And they didn't seem to have, near as I can tell, expertise that would have allowed them to recognize that this was a new product and they really needed to do something more than the usual vaccine testing. And there was a number of things in that dossier that, as an expert in the field, that kind of developed most of the core platform tech, including the use of luciferase in animals, which is what they were using. They never actually tested these RNAs before they went into the clinic. They tested a RNA encoding for a protein called luciferase, an unfortunate name. It has nothing to do with the devil. It's the protein that causes the firefly tail to glow. And Pfizer did a really interesting trick. they used to look at the biodistribution, where the stuff goes and where it produces protein. The rigorous way to do it is you take samples of tissue from the mouse or the rat or whatever you've done the delivery into for this animal model, and you analyze each one of them in a tube in a very, very sensitive assay. Then there's kind of a parlor trick, if you want to get your picture on the cover of the Rolling Stone, no, of Cell magazine or whatever, in which you can take the whole animal and deliver the luciferase gene or RNA, causing it to make the firefly protein, and then inject it with the molecule that the firefly uses to make light, and put up a real sensitive camera at a full photomultiplier tube. And you can actually see the photons come through the animal.
Really?
Okay, it's cool, right? So, and so you get a picture of an animal with little glowing patches, and you can say, oh, that's the liver there, and that's the bone marrow, and whatever. This is the least sensitive way to detect where the RNA would have gone and express the protein, because those photons have got to come up through the skin and the tissue and the muscle and everything else. They get scattered all over the place. So Pfizer used the least sensitive way to detect the biodistribution of the RNAs and where it's expressed in the animal model, and the FDA didn't catch them at it. Okay, and when I saw that, I knew that we were in trouble. That whoever was, watching the store had no idea what they were doing, and they were having the, getting back to common metaphors, they were having the wool pulled over their eyes by the pharmaceutical industry. And I think that's pretty much what happened. And now we have all these things coming out about integration and reverse transcription, and well, those are genotoxicity things. There were no genotoxicity studies done. There were no rigorous, good laboratory practice controlled, which is the standard, reproductive toxicology studies done. And now we hear all these things about women with problems with their periods, and there is signal, let's say, about spontaneous abortions and fetal deaths and all kinds of things that have to do with reproductive health. All that should have been detected long before it was ever put in humans. But the FDA didn't force the pharmaceutical industry to do its job.
Well, it's ironic because you mentioned those documents which are now being released, the ones that they were trying to have to take 7 years to release. Yeah. But the latest batch of documents, it mentioned there was a consent form, and people had to specifically sign off saying, I acknowledge that this might have some fertility issues in the future. So Pfizer did have at least enough foresight to know that legally they have to cover their bases to say that this might be the case. And probably in the trial, there was not enough time to determine whether it was the case or not. And so one of the things that a lot of people have been asking me to ask you, once I get a chance to sit down with you, is that, let's say a year, 2 years, 3 years down the line, besides this mRNA that stays in the body for a longer period of time than expected, what else can people foresee down the road could be potential long-term side effects of this vaccine? Or is it the case that everything does leave the body eventually and it kind of restores back to normal?
So that's what you're asking me to do is look into my crystal ball and say what's things going to look like 3 years from now. Which, by the way, has been what I was, one of the people attacking me in the New York Times criticized me for doing that specifically, projecting or anticipating what the results were based on current data and where things were going to go, and that this was unethical that I'd done that, even though I had been right in so many things. My point of view on that is, well, yeah, so I was able to interpret the data better than you, and you're complaining about it. Be that as it may, this particular critic had about 1/10 of the citations that I've had in my publication record, so really a fairly junior faculty member. But so I have had in this technology area a pretty good track record of calling it before it's obvious to many people. Like, for instance, when I went on the Laura Ingraham show and said that it looked to me like Omicron might be a Christmas gift, a lot of people found that kind of shocking. But that's turned out to be the case. At the time, we had the Biden administration saying it was going to be a dark, horrible winter of death for people who were unvaccinated. That didn't pan out, right? So with that background, what do I see in my crystal ball, knowing that I'm far from perfect? So these are speculative risks, let's say, that should be mitigated. That's the way I like to think about things. The data are getting stronger that the multiple vaccinated have a higher risk of infection and significant disease and death from Omicron. What happened before with Delta and the prior variants is kind of irrelevant now. Now we have a mismatched vaccine that is being administered repeatedly, and there are multiple lines of evidence demonstrating that can cause immunosuppression, and people are talking about AIDS. There's two aspects to this AIDS story with these RNAs. One is that there are short sequences in the spike protein. Remember, this is the spike protein that was engineered by the bat lady in Wuhan, as far as I'm concerned. And so there are short sequences there that have what we call homology or similarity to short sequences in the AIDS virus. That doesn't mean they were pulled from the AIDS virus. Viruses have convergent evolution. They have a lot of overlap. There's only so many ways you can solve a biologic problem. Okay, but there are these sequences that have some similarity to AIDS sequences, and they can cause false positives in HIV testing. So people kind of get wrapped around the axle thinking, oh, they put AIDS virus sequences into the vaccine, and the vaccine can cause you to get HIV. That's not what those data suggest. It's a cross-reactivity. It's a form of artifact. However, AIDS is an acronym. It's Acquired Immunodeficiency Syndrome. That's what it stands for, Acquired Immunodeficiency Syndrome. And the data are getting stronger and stronger that the multiple vaccine recipients are having, as we mentioned before, a T cell suppression. Their T cells aren't functioning as well as they should be. And you can see various markers of downregulation of key signaling molecules and the nuances that we don't have to go into. It's measurable. Okay, so there is evidence of an acquired T cell immunodeficiency in the multiple vaccinated. Okay, so is that clinically significant? You always got to say, well, you can measure something in the laboratory, who cares? Is it actually causing problems for patients? Well, Ryan Cole was one of the first ones to signal about the canary in the coal mine of the appearance that there are cancers springing up that are unusually aggressive at early age, or that were in a kind of an indolent or slow growing stage and then suddenly become very aggressive cancers. He's talked about endometrial cancer originally, cancer of the lining of the uterus. But there's many others now that are popping up that are being observed by cancer surgeons and diagnostic radiologists and pathologists. People often, when they attack Ryan Cole or myself, people that are trained in pathology, they don't recognize that we're not like average docs. We're trained in signal detection. That's what we do. The discipline of pathology is the quality control for the entire medical care system, and that's why we have to do autopsies. It's not pleasant, I'll tell you. Nobody takes pleasure in doing autopsies, at least nobody I know. But it's the QC, and pathologists like Ryan and I are trained to detect patterns. So Ryan early on detected these patterns about what appears to be unusual cancers that are unusually aggressive at an unusual frequency. That seems to be playing out. So that's why would that happen? Well, one of the things that keeps cancer in check is your T cells. We all have cancer rising in our body in various points in time. And so seeing these signals is another data point along that line saying, T cell suppression. There's another one. A lot of these people that have the post-vaccination syndrome, which is indistinguishable in symptoms from long COVID. Okay, people with post-vaccination syndrome, this chronic malaise, weakness, lack of energy, sometimes sleepiness, other things like that. Many of them have what's called reactivated latent DNA viruses. Or another thing that was known very early on, we all talk, the CDC has acknowledged the myocarditis. The same team that identified the myocarditis, which by the way was outside of the review branch of the FDA. It wasn't people at CDC. Okay, people outside the review branch of the FDA detected the myocarditis, and that set a cascade off. Another thing they detected really early on was the unusual incidence of shingles, reactivation of a herpes family virus from people that previously had chickenpox, and which was residing in your neurons. That's what that virus does. And it's kept in its Pandora's box by your T cells. So when you're seeing this unusual incidence of shingles in adults after vaccination, well, that was one of the early warning signs about suppression of T responses. Now, what Richard also just disclosed, and many other docs are seeing in his talk just today, is that we're seeing reactivation of Epstein-Barr virus. Okay, so this is kind of the infectious mononucleosis, chronic malaise, fatigue, etc. That seems to be very common. And again, the problem, so there's CMV, herpes, shingles, Epstein-Barr virus. These are all latent DNA viruses that are kept in their box by the T function. And so seeing all these things pop up is another sign. So immunosuppression, cancer, reactivation of latent DNA viruses, more and more signals coming up about reproductive effects. And those are perhaps among the most worrisome. And of course, we knew from the original Pfizer data in that common technical document that the lipid part, the positively charged fats that wrap the RNA that allow it to slip into cells, they are, in the limited studies that Pfizer did, they accumulated to a very significant level, an unusual level, compared, say, to testis or other organs. They seem to be concentrated in the ovaries. And so that, the ovaries are what drive the function of the uterus. The ovaries are what drive menstruation. So we had, concurrently all these reports coming from women saying, hey, this is really screwing up my menstrual cycle. And we saw that in the Orthodox Jew population in New York when I gave testimony there, because the rabbis closely monitor menstruation. It's key in their religion, having to do with reproductive practices, which is really important to rabbis in the Orthodox Jewish community. And they were seeing this. So, but the CDC and the scientific press in general denied this, and they asserted that this was, I still can't believe it, female hysteria. This is such a mid-century concept that women are being hysterical. I mean, the root of the word hysteria is uterus. It comes from the Greek for uterus, hyster. Okay, and so it was being asserted by the CDC and the medical community that all these women reporting these variations in their menstrual cycles, in some of them onset of menses in the postmenopausal women, that's often a sign of cancer, and long delays in menstruation. All these things are pointers that there's something going wrong with the ovaries, because the ovaries drive menstruation. And so then we have this collusion of facts that the lipids seem to go to the ovaries. They're a synthetic abnormal lipid. None of the lipids in your bodies are positively charged. They're getting concentrated there. Something's happening, and no one's interested in investigating it. But nevertheless, it's being observed. So there's a bunch of reproductive effects. There's signal coming out now about elevated spontaneous abortions, fetal death, and even things like in the nursing a female child, passing blood clots per vaginum, highly, highly unusual. So there's a cluster of things about reproductive health. Then we have this myocarditis problem. And again, there was a strong concerted effort by propaganda, censorship, etc., to deny the risks of myocarditis and to downplay them. But the incidence of clinical myocarditis in Hong Kong in the most comprehensive study, we're talking about little boys going to the hospital after getting their vaccine because they've had damage to their heart. That incidence was 1 in 2,700. That's not nothing. And there's all this talk, oh, it's mild myocarditis that goes away. No, there is no such thing as mild myocarditis. The heart does not heal. That's why there's all this focus on myocardial stem cells, is the heart doesn't heal, it scars. And when it scars, it forms little areas that are electrically irregular. And the heart is not just a muscle, it's also an electrical organ. That electricity and how it circulates is crucial for the heart to contract in a harmonized way that makes it effective. And if you have little areas of scars in your heart, this can cause irregularities in electrical transmission, and that can set off things like ventricular or atrial fibrillation, particularly ventricular fibrillation, which causes sudden death. And if you have small scars and you have somebody under high performance stress, let's say a high performance athlete functioning at the peak of their capability, they can suddenly get ventricular fibrillation and drop dead. Yeah. What are we observing all over the world in unusual numbers is death of high performance athletes and even people up in their 40s and 50s that shouldn't be dying of heart attacks, and they're dying at unusual rates. So I think we are going to see the long-term consequences of this myocarditis. You're asking me to list things. Mm-hmm. And then we have these various blood clot problems because we know that the spike protein is causing blood clotting, and it also appears to be causing some autoimmune problems in some people, like autoimmune problems that attack blood platelets. And so this can cause hemorrhage, clotting, and other things. And this purpura, it's called, where you basically develop bruises all over your body when you have low blood platelets. And this is a consequence, it appears, of the spike protein binding to platelets, and then your body seeing that as something different and new that it should attack with antibodies. And this is known as one of the causes of this autoimmune thrombocytopenic purpura, which does seem to be occurring at a reasonable rate. So over the horizon of 2 to 3 years, I think it's likely we're going to see other forms of autoimmune disease. Wow. So there's a brief tour. For more information, I refer you to the 9 pages of adverse events revealed in the first data drop in that table from Pfizer. That's
right. It was 9 pages, single-spaced, separated only by commas. And actually, so along the lines of what you said, there was a recent report released, I believe it was the mortality report by the CDC, which found that Americans who are in the prime of their lives, meaning 18 to 50, the rise in deaths among those people, non-COVID deaths, has spiked in the year 2020.
So, yeah, double entendre.
It's, it seems to fit in exactly what you're saying.
So this is, my friend Ed Dowd has been one of the leading voices in this. Ed is former BlackRock stock analyst. And I met him actually last fall in Maui when we did one of the first global COVID summit interventions in Maui. And by the way, it was in support of Kirk Milhound and Lauren Pang, people that I mentioned on the Rogan show, and that caused a certain kerfuffle in Maui. But those two gentlemen were both being attacked for providing early treatment or recommending against vaccination in pregnancy. And people tried to take away their license. It's just the last week that they've finally overcome that, and their licensing board has decided not to take any action against them. And of course, Kirk is here at the rally today. So I met Ed at Maui when we were there last fall, and we had a number of discussions about the importance of integrity and the loss of integrity throughout our government, and particularly the CDC and the FDA, and what that means. And he came up with something called the Malone Doctrine. That's a fascinating document that's very much focused on the need to restore integrity. But he's taken it further. He was the one that first really picked up and recognized the importance of the report from this Midwestern multi-billion-dollar life insurance company that was primarily insuring employed persons, that showed this spike in death that corresponded, it followed the spike in vaccination, mandated vaccination. And then he's continued to pursue this with the insurance industry. And the insurance, as the data have come out, because the insurance industry that is publicly traded have to reveal key actuarial data with their quarterly reports. And so he's tracked this carefully, and as you point out now, if you parse the data by age and plot it over time, what you see is a series of rises and falls in all-cause mortality, which in the end, because there's been so much data manipulation by the CDC and withholding data, it's like the only valid indicator that we can use right now is all-cause mortality. And these spikes in all-cause mortality, again and again, correspond with spikes in vaccine uptake in countries all over the world. They are way off scale in terms of the norms in changes in all-cause mortality that's been seen historically. And so the insurance industry is picking it up, and they're also having to pick up the tab. And it's looking like it's going to have a significant impact on their bottom line and on their actuarial tables. And these are data that can't be manipulated and hidden by a federal agency. As you point out, all-cause mortality is the ultimate outcome and has always been a required outcome in any clinical trial, is all-cause mortality, for the reason that sometimes you have adverse events that you don't think of, and you say, oh, it couldn't possibly be related, Oh, he got hit by a truck. Well, that's not vaccine related, right? Well, what if the vaccine has caused somebody to have mental problems, brain fog, and they happen to walk across the crosswalk and and subject themselves to greater risk, or when they're driving, you would say, oh, they got hit by a car, that's not vaccine related, when in fact it could be vaccine related. So my point is only that all-cause mortality is the one variable that you can measure in any clinical research, and should, because it doesn't carry the bias of causation.
That's a great point. I know I've kept you for a very long time, but I have one more question. With the latest data drop from the FDA, one of the things that I believe is really worth highlighting is the fact that even in the initial Pfizer trials, they were very much aware that natural immunity was effective.
Yes.
Very, very effective at preventing severe disease.
And yet it was suppressed.
However, the FDA and the CDC, in theory, at least that's what they said, they went through this data with a fine-tooth comb, right, in order to approve the Pfizer vaccine. And so they should have been aware of
that is their public position that they did so.
Yeah. So why? Why do you think that's the case, that they suppressed natural immunity?
I'm told that there's in the written record statements from people that were present but remain anonymous, that there was a group of 4 individuals that took up this issue. And one of them has come out saying that he was a strong advocate for recognizing natural immunity, but that he was overruled by the other participants in this small group, which one would assume at the time would include Deborah Birx, Tony Fauci, and Francis Collins. So it appears that there was a decision on the part of key leadership in Health and Human Services in the United States government to suppress that information and to deny the obvious, which is that the infection recovered individuals have equal or superior immune responses to those that have been vaccinated. This has always been the case. And then 2 or 3 weeks ago, there was a fascinating paper dropped from Sweden, which you may or may not have followed. This was the one that talked about the IgA and the importance of IgA versus IgG. And what this paper showed, and it was data that had been captured very early on, before there were any vaccines, in healthcare workers, so people subjected to a lot of virus all the time. And there's a cohort there that never got infected, and yet they were they were exposed to virus all the time from infected humans as a course of their work. And they had no other alternative, and yet they never came down with disease or any sign of infection. It turns out that this population is highly enriched in IgA, which is the secretory antibody that's produced in your nasopharynx and your gut and And antibodies, people think of the immune response as all being the same all across your body. It's not. B cells and T cells home to specific places, and they basically encounter antigen and get educated in various, this is the terms we use, in lymph nodes or thymus, and then they go back to those same places, okay? They have homing. They have recognition signals that allow them to home back to where they're supposed to go. And when you have natural infection, it's in your nose and your mouth and your upper airway, and initially, and it evokes what are called secretory IgA or mucosal immune responses. And it appears that one interpretation of these Swedish data is that these people were constantly getting exposed to low levels of virus from their work environment, and it was eliciting a IgA predominant mucosal immune response, which is highly protective, and kept them completely out of the hospital or out of their bed rest for COVID. Whereas those that had predominantly an IgG response, which is what is elicited when you give the jab, actually had higher risk. So this again, there's all kinds of lines of evidence that this overlooking, let's make it as benign as possible, or intentional suppression, that's the worst possible scenario, of recognition of the importance of natural immunity, was a major error in management of this outbreak, and there have been many major errors, but that may be one of the more egregious ones.
Yeah. Dr. Malone, I'd love to speak with you more, but I think that's all our time for today. Thank you so much.